Hello everybody, good afternoon, hope you are well. So welcome back to Understanding Cancer Season 2, and today we're going to be doing Lecture 6 titled Types of Signaling Pathway, Normal and Dysregulated GPCR. So hopefully from last week you should have understood that the first messenger in the cell signaling pathway is the ligand. Okay. The secondary messenger is involved in a signal transduction, so that is necessary in order to perform a cellular response. Calcium ion channels are found in organelles, which are little organs found in cells, such as mitochondria, endoplasmic reticulum, and plasma membrane. It is regulated to maintain its low concentrations in cells. It binds specifically to the calcium binding protein calmodulin. CAMP plays a key role in signal transduction pathway. It is activated by adenyl cyclase, where it then binds to protein kinase A to activate more proteins in the signal cascade. CAMP is deactivated by phosphodiesterase. We also looked at other pathways that involve signal transduction and cellular response, for example, deacylglycerol and inositol triphosphate. So they are also secondary messengers, and they phosphorylate downstream proteins and also stimulate other secondary messengers such as calcium ions, respectively. There is also transcription factors. They regulate genes, the production of genes, and this influences the cellular response. So what will we learn today? So hopefully we will learn or recap the structure of the G protein coupled receptor in a in-depth way, look at the subtypes of the GPCR, and we will also be looking at the three key steps, so receptor activation, signal transduction, and cellular response that occurs within the normal GPCR signaling pathway. So in the previous episodes, we have briefly looked at, generally speaking, about the structure of the GPCR, how it gets activated, ways of it gets, the signal can get transduced, and also the cellular response. However, this is a more in-depth of understanding and with regards to its structure and how it performs its action. We'll also be looking at how to turn off the GPCR signaling pathway and the causes of why GPCR gets dysregulated. So why does it stop working properly? And we're going to provide some examples of cancers where this is present. So a gentle reminder, an ideal way of learning is to divide the time across the 7 days. Okay, so even with the preparation of these, I'd normally divide it over the few days or around a week and so. So approximately this lecture takes about 1 hour maximum. However, if you want to divide this 1 hour across the different days, then please proceed and challenge yourself with a quiz afterwards. Just to recap, to support your learning, this lecture and all lectures, they have diagrams that are designed by HN Designs unless otherwise specified, and it's presented in a simplified way. There is a glossary to help understand what keywords mean. There's also summary doodle revision posters to assist with your learning, and there's quizzes to test your understanding, and also there's a reference list for further reading. So I want to thank a special thank, especially my parents, my family, my friends, also my lecturers, because initially how I was introduced in terms of cancer signaling pathways and all the diagnosis and treatment is through my lecturers and also respected health professions who taught me and installed the passion. So thank you to them too. So let's begin with the structure of the G protein coupled receptor. So far you should have understood when we looked at the lecture in receptor activation that GPCR is a helical transmembrane receptor protein that is found on the cell surface. So you have some receptors that are found inside the cell, and you also have some receptors that are found on the cell surface. Now the GPCR genes, they account for 5% of the human genome. So the cell surface receptor is divided into 3 regions. So the 3 regions are as follows: extracellular loop region, the transmembrane region, and the intracellular membrane loop. So what occurs in the extracellular loop region? Well, this is where binding occurs to the specific ligand. Okay, so the first messenger. Now the transmembrane region is what is embedded within the cell membrane, and it comprises of 7 transmembrane proteins, and they are helical shape, and they're like the middle factor where the intracellular, which is inside the cell, and extracellular, which is outside the cell, they communicate together. So this is where the changes occurs. Okay. Now the intracellular membrane loop, this is where more or less interaction with the intracellular proteins called G proteins takes place. So this is the part where the signal is transduced inside the cell, and then it begins its journey with the phosphorylation and the cascade. So in the previous lecture, this is the structure that the GPCR receptor is presented. So you can see there's the ligand binding site, you've got the 7 helical proteins there, you've got the plasma membrane, and also attached to the GPCR is the G protein. So the G protein in an inactive state, you have the energy source guanosine and diphosphate, GDP, and you also have the structure of the alpha subunit and the beta and the gamma dimer. Okay, so the G protein consists of, in an inactive state, the GDP, as well as the alpha subunit and the beta gamma dimer. Okay, you can see that there is loops within extracellular, and there is also loops within the intracellular. So some members of the rhodopsin family may have 8 helices. So instead of having 7 helices, some actually have 8 helices and 4 intracellular loops. So what are the subtypes of GPCR? So we've mentioned one of them, which is rhodopsin. So that has 8 helices and 4 intracellular loops. The other members are adhesion, glutamate or glutamic acid. You have secretin, and you have phosphatase2 and protease activated receptors, PARs. So now you can see that within these loops there are categorized. So you can see there's E1, E2, E3, E4, and then you've got the 7 transmembrane regions, so H1, H2, H3, H4, H5, H6, H7. And then the intracellular membrane loop, you've got C1, C2, C3, and C4. Now, the purpose of why you have the NH3 plus and the COO minus is because that the GPCR is a receptor, and a receptor is a type of protein. And the structure of the protein comprises of amino acids. And these amino acids, they have an amine group, and they also have a carboxylic acid group. So I'm going to be mentioning some of the functions of some of these structures here. So C3, as you can see, is part of the intracellular membrane. So in that part, that's where it interacts with the G proteins. And there's also sites called phosphorylation sites. So there's the protein kinase A phosphorylation sites there. And you can see that's apparent in C3 and C4. Now C4, in addition to that, it has G protein coupled receptor kinase phosphorylation sites. So a gentle reminder that the kinases are a type of enzyme that basically add a phosphate group to their target or substrate they act upon. And it also interacts with G proteins. Now E4, that's part of the extracellular loop region. That has the messenger binding site in the roots. That's where the ligand actually binds. So let's look at or have a recap on receptor activation of GPCR. So in a previous episode, we discussed that the ligand binds to the GPCR, and this is what causes a conformational change. So let's look at examples of ligands. So you have amino acids, you have also hormones, neurotransmitters. So these are chemicals that are involved in transferring the messages between a nerve and another nerve. You also have chemokines, which are a type of proteins involved in the immune system that helps to protect the body from an infection. And you also have other proteins and small ions. So when the conformational change occurs, this is what leads to the interaction with the G proteins. So in the first image, you can see there's the ligand there, but it's an inactive receptor because there's no binding occurs. But in the second image, you can see that the ligand has bound, and now that's why it's activated. So let's have a recap of what we understood so far. So in the previous episode, what you should have understood is that you have a guanosine diphosphate, which consists of two phosphate groups, a sugar, and a nitrogenous base. So in this case, guanine. This is why we've got guanosine. And it's attached to an alpha subunit and a beta and a gamma dimer. To build upon this, what we can understand is that a G protein is known as a heterotrimer because it consists of those three subunits. And they also are classified based on the alpha subunits. So you have a G protein, but you got different types of G proteins. So you got a G alpha I and a G alpha S, the G alpha 12/13, and you also have the G alpha Q /11. And the G alpha I and the G alpha S, they regulate adenyl cyclase activity. So let's have a recap of adenyl cyclase. So in the previous episodes, we discussed that this is what is involved in the production of the CAMP, which is cyclic adenosine monophosphate. And CAMP is an example of a secondary messenger. And it utilizes ATP, which is an energy source, in order to produce this. And ATP is an abbreviation for adenosine triphosphate because you've got three phosphates and a sugar and a nitrogenous base. So how do the G alpha S and G alpha I regulate? So if you look at the image over here, the G alpha I is involved in halting the activity of adenyl cyclase activity. So what that means is it stops the production of the CAMP or lowers the activity. But with the GAS alpha S, what the alpha S is involved is to activate the adenyl cyclase activity. So you have more of a production of the CAMP. The G alpha 12/13, that's involved in the pathway that involves row A, whereas within G-alpha Q11. So the GTPase binding domain, it exchanges the GTP for the GTP. It has 3 switch regions that are flexible loops, and this is what changes the conformational when it's bound with GTP. And it also has the ability to bind to the G, beta, gamma subunits. Okay, and it's also involved in binding to the GPCR2. Now the helical domain, that is involved in the or has 6 alpha helices that has nucleotides in the protein core. Now, once the activated receptor activates the G protein and then exchanges the GDP to GTP, the ligand has done its job, so the levels is what decreases, because now it's been activated. You don't really need the ligand to be present there when you've got the G proteins already activated. So this is an image of what an active G protein looks like and an inactive G protein looks like. So in a active state, you can see that you have the 3 phosphate groups, the sugar and the guanine, as well as the alpha, beta, gamma dimer. Okay, but in an inactive state, you can see the presence of 2 phosphate groups, a sugar and a guanine. So let's proceed on to the signal transduction. So the alpha subunits from the G protein is what binds to the enzyme adenyl cyclase on the plasma membrane. So when the G protein is activated, the alpha subunit and the beta gamma dimer, they don't stay together, they separate, they dissociate. So what you have is the G protein being activated with the alpha subunit, and that is what attaches to the enzyme that is responsible in the production of CAMP, secondary messenger. And this is achieved via ATP, but there are also other targets for CAMP. So you have EPAC2, EPAC1, and nucleotide gated ion channels. So this is an overview and then over here, just to simplify it, the CAMP depends on 2 types of enzymes. You've got adenyl cyclases and phosphodiesterases, PDE. So the adenyl cyclases is involved in the production of CAMP via ATP, whereas the phosphodiesterase, that is what lowers the levels of CAMP. And what it does, it converts the CAMP to AMP, which is adenosine monophosphate. And currently, according to research, there's about 22 PDEs that has been identified. So let's have an insight in terms of the types of CAMP targets. So EPAC is an example of a guanosine exchange factor, and it has a CAMP binding domain. Okay, so this exposes the active site for the catalytic domain. So this image is from Creative Commons. So what you can see over here is you've got the GPCR within the plasma membrane. Then you've got the G protein attached to the GPCR. When it's activated, the G alpha is attached to the adenyl cyclase that is involved in the production of CAMP via ATP. Now, one of the targets of CAMP is EPAC1-2. So that converts the downstream target, which is RAP1/2, that is attached to GDP, and then it phosphorylates it by attaching another a phosphate, so it becomes RAP1/2 GTP, and it further leads to downstream signaling targets. Now, in part B, if you look at the different structures or types of the EPAC, you have a regulatory region. So that regulates the activity of the catalytic region. The catalytic region is what contains the active site where the substrate binds to the EPAC. So if we look at the first one, EPAC 2A1, this you can see there's 2 camp binding domains over there. So you've got camp A and then camp B. And then the EPAC 2A2, you also have these binding domains of CAMP. EPAC 2B, you've got the CAMP-B only. The EPAC 2C, similar the case with 1 CAMP B, and EPAC 1, you've also got the CAMP B. Yep, so EPAC 2A1 and EPAC 2A2, those are the only 2 that have 2 camp binding domains in the regulatory region, whereas the remainder types of EPAC, they have 1. So that the purpose of the camp binding domain is involved in activation of the EPAC itself. The DEP region that is found in the regulatory side of the EPAC, that's involved in localization. And you have the REM, that's involved in the exchange between GDP to GTP. And you have the RA, that's interacts with GTP RAS, which is a type of oncogene, which we'll discuss in another type of signaling pathway. And you also have CDC25HD, that is present within the catalytic region, and that's involved in the guanosine exchange factor activity. Another type of CAMP target is cyclic nucleotide gated ion channels. And these are channels that allows positive ions, known as cations, to pass through. And this is what changes the membrane potential, so the concentration that's inside and outside of the cell. And CNG is an abbreviation for cyclic nucleotide gated channels, and hyperpolarized activated cyclic nucleotide modulated HCN channels. So as you can see over here, that within the CNG, it focuses on the potassium ions that is more or less leaving the cell, and the HCN is involved in ions such as sodium and calcium that are entering the cell. However, the focus of today is protein kinase A. So this is one of the CAMP targets which we will focus on. So pKa is a tetramer enzyme. Okay, so it comprises of 4 subunits, and it can be classified into type 1, type 2. So in the previous episode, we discussed that the protein kinase A is what binds to the CAMP, and that's what leads to more of the signal cascade taking place. So the pKa type 1, that is found within a cytoplasm, and it consists of Ri alpha 2C2, RI beta 2C2. Whereas the pKa type 2, that consists of the R2 alpha 2C2 and R2 beta 2C2. And so the pKa type 2, this is found within the structures. So pKa is a tetramer that is divided into 4 subunits. So 2 catalytic subunits and 2 regulatory subunits. So what that means is you have 2 catalytic subunits together and 2 regulatory subunits together, and they're all combined together to make 4 subunits. And within mammals, there are 4 types of regulatory subunits: RIA alpha, RI beta, R2 alpha, R2 beta. And there are 3 types of catalytic subunits, so C alpha, C beta, and C gamma. So this is some interesting findings to find out that within the mammals they have further types. So in the location of where the regulatory subunits can be found, RI beta can be found within the brain, testis, and beta lymphocytes, which is a type of white blood cell. And RI alpha and R2 alpha are found in a variety of different cells. And R2 beta is expressed within the brain, fat, and some endocrine tissues. So here you can see the 4 subunits attached together, and the regulatory subunit is what has the camp binding sites. So you can see within the structure, you can see some camp binding attached to it. So the purpose of the catalytic subunit is that they phosphorylate specific cellular proteins. And the regulatory subunits, what they do, they bind together in order to start the activity that is involved within the catalytic subunit. So the regulatory subunits is what initiates the activity for the catalytic. And the CAMP binds to the regulatory subunits of pKa. And when it activates the catalytic subunits, they separate array. So in an active pKa, this is how it states. But when the CAMP binds onto the regulatory subunit, it leads to the separation between the regulatory subunit and the catalytic subunit. So that's the active form of protein kinase A. So the activated catalytic subunit of pKa, they phosphorylate specific cellular proteins, and this is via the amino acids serine and threonine. So the proteins are made up of building blocks, which are known as amino acids, and there's a variety of different types of amino acids, and 2 of these types is where phosphorylation takes place. And examples of cellular proteins are enzymes, such as glycogen synthase kinase. There's also RAF, structural proteins, BAD, that's involved in apoptosis. Also transcription factors, so those that are proteins that regulate the gene transcription, such as CREB, so camp responsive elements. So there was some interesting findings that I wanted to share with you all. Is that the recent study one is that pKa is an actin-myosin contractility regulated effector, so it's involved in cell migration. And in the previous sessions, we discussed about actin, filaments, myosin, and intermediates. So these are part of the cytoskeleton that helps the cell to migrate. Then you have recent study 2, where pKa has the ability to phosphorylate this type of protein called CDC42, interacting protein CIP4. And that basically facilitates how the change in the shape of the membrane, and also how the actin polymerizes. So when you have lots of small molecules called monomers, when they bind together, it forms a polymer. So how the actin protein polymerizes to form a larger polymer, the CYP4 facilitates this. And this is what helps in terms of migration and invasion and metastasis of cancer. And recent study 3 is that activated pKa can inhibit adenyl cyclase, particularly subtypes AC5 and AC6. And this is what activates the phosphodiesterase PDE3, PDE4, and which lowers the levels of CAMP. And there's an interesting finding is that you have pKa anchored proteins, which is called AKAPS. And what they do, they bind to proteins that are involved in the cytoskeleton, such as actin, myosin, and they also bind onto organelles and to the pK regulatory subunits. So there's the AKAP, they have an additional binding site, and where they can attach to the pK regulatory subunits and provide an additional site for substrates to bind. So one of the targets for pKa is CREB. So CREB is a type of transcription factor, and it's a member of a basic leucine zipper, BZIP, superfamily transcription factors. And it can be found in many types of tissues. There are other types of transcription factors that belong to the superfamily there. That's called CREM. That's also found in many tissues. So you've got a CAMP responsive element regulatory protein. And you also have transcriptional activator 1, ATF1, that's found in neuroendocrine tissues. So they can form homodimers, so two transcription factors the same, or heterodimers different. But what's common in all of these transcription factors in this superfamily is that they have a KID, which is called a kinase inducible domain, and that's made up of 60 amino acids, and it's located within the central region and contains the pKa phosphorylation sites called RRPSY. So this is mainly for cyclins, apoptotic proteins, growth factors, enzymes, and transcription factors. So here is an image of the CREB. So you've got the KID that is found in the middle there. Then you've got Q1 and Q2. These are two types of amino acids called glutamic acid or glutamate. And you also have the BZIP, which is the basic leucine zipper domain. So that is what characterizes all of the transcription factor in which CREB belongs to. So let's have a look at an example of what happens when CREB becomes activated. So CREB can be phosphorylated via the amino acid called serine 133. And another transcription factor can be activated, ATF1, via SER63, so serine 63. And not only can it be activated by pKa, but also AKT, RSK, MSK, CAMP-K2, and CAMP-KIV. So at the moment, I don't want to go in depth of each of these because there will be signaling pathway lectures which we're going to be discussing later on, where we will go more in depth. But just giving you a brief idea that pK isn't the only enzyme in which the CREB ATF can be activated by. So the ATF and the CREB can form a homodimer, i.e. you can have two ATF1 and two CREB, or they can form a heterodimer, which involves ATF1 and CRAB. And what happens is it binds to the CAMP responsive elements called CRA, and that's found in the promoter region of target genes. So the promoter is the area where transcription takes place, so the starting position or the initiation site. And you can regulate transcription of target genes to initiate the cellular response, for example, oncogenes such as CGen and cyclin D1. So G protein independent pathway. So there is also, you can activate the adenyl cyclase, which is involved in production of CAMP without the use of the G proteins. And this can be achieved by calcium ions and bicarbonate ions. So let's look at the cellular response. So cellular response involves different cellular activities, for example, cell proliferation, so in how it grows, how it survives, how it moves, the energy that is involved, how it differentiates into more specialized cells, and the sensory perception. Hematopoiesis, this is the production of blood cells, okay, and this is what occurs within the bone marrow that's found within inside the bone. There's also other cellular activity, for example, the transmission of nerve impulses, immune response, apoptosis, and glucose homeostasis, and also memory and learning. So how does the GPCR signaling pathway turn off? So this can be achieved whereby the ligand dissociates from the receptor which inactivates GPCR. Therefore, the ligand degrades. The alpha subunit of the G protein hydrolyzes GTP to form GDP and P, which is the phosphate. The alpha subunit and the beta gamma dimer join back together and become an inactive G protein, and the levels of the CAMP decrease because of the enzyme phosphodiesterase. So the phosphodiesterase is what hydrolyzes CAMP to form AMP. And then the low levels of the CAMP causes the regulatory subunit of pKa to release CAMP. Now the 0.16 is that the regulatory and catalytic subunit of pKa reassociate together, because you said that the activated form of the enzyme is when the regulatory and the catalytic subunits separate. But when they both reassociate, they become an inactive state. And this is stimulated by enzymes called protein phosphatases. So the GPCR signaling pathway is involved in the cancer growth and development, where it's involved in a number of hallmarks of cancer, such as unregulated growth, invasion, metastasis, evading apoptosis, also evading the immune response, and angiogenesis. So what are the causes of this dysregulation to take place in the first place? Well, just to identify that the dysregulated GPCR signaling pathway is involved not only in cancer, not only in asthma, but also in heart disease. And there's a number of causes such as point mutation, increased production of the ligands, overexpression of copies of the GPCR gene, silencing of the GPCR via the promoter methylation, so that's the addition of the CH3 group attached to it. And you also have mutant forms of G proteins, so mutant proteins. So what is a point mutation? So a point mutation is changes to the DNA sequences, but has no effect on the amino acid and the protein. So this type of point mutation is a silent mutation. But there is other types of point mutations that do have an effect. For example, missense, the DNA sequence has been changed because there's a substitution. In other words, the base is different, and this causes a different amino acid to be made. Okay, and because the amino acid is formed by a codon which consists of 3 bases, and it's very specific. So if you have a change within the codon which consists of the 3 bases, so you change one of the bases, that changes the amino acid, because these codons are very specific for these amino acids, and in turn that affects the production of the protein, whereby the shape is changed and also the function. There also is a change within the codon to create a stop codon, so it's not finished. So this is nonsense, and this affects the structure of the protein, because instead of completing the whole protein, it forms a short protein, and this can either be functional, where it works, or the protein doesn't work. There's also the splice site, so this affects the mRNA template, because we discussed that when the DNA is converted into RNA in the transcription stage to produce the specific protein in question, the RNA has a sequence where some of it do have the sequence that codes the specific protein, and these coding regions are known as the exons, and you have some that are irrelevant, which are the introns. So this is why it needs to get spliced, where you only need the template that has the sequence necessary to produce a target protein, rather than having additional sites or regions that doesn't code for anything. And when in the stage where you've got a point mutation, what happens is some of the exons that has the coding or the templates for that particular protein are removed, and the introns that doesn't mean anything is added. So it forms a different protein than the target protein that you want to produce. So this image is from Creative Commons, and what you can see is in the structure there, the codon is made up of 3 bases. So this is AAG, so it's adenine, adenine, guanine, and then in the mRNA stage it's AAG, and in a protein it forms the lysine amino acid is what forms part of the protein. But when you actually have a point mutation taking place, you can see that in the DNA stage you have AAA, and the mRNA, instead of having AAG, you have AAA, but it still produces the protein lysine, so that's more of like a silent form. But when you have in a DNA where you've got the TAG, and the mRNA stage has UAG, and then it performs like a stop codon, so it shortens the protein. And the reason why it's UAG is because when we look at the structure of RNA and DNA, one of the differences between DNA and RNA is one of the bases, instead of called thymine, is uracil. So the UAG, it's still the same there, but it formed a shorter protein, so that's the issue there. And in the third instance, you've got the AGG instead of the AAG, and that is what's being produced within the mRNA template, and it forms a whole different type of amino acid called arginine. So that affects the protein structure. And then in another example you have ACG. Now ACG, you can see that's not the same as AAT, and as a result you're producing another type of protein called threonine. So you can see there's different types of point mutations there. Now, the activation of the alpha subunit of G proteins, this can be done via chemokines. So these are type of proteins involved in the immune system, such as interleukin. You also have neuropeptides, such as prostaglandin. So neuropeptides, these are peptides that are produced within the nervous tissue in the central nervous system and the peripheral nervous system. You also have lipids such as lysophosphatidyl acid. So this is also found within the nervous tissue. So all of these factors, when there's an activation of the alpha subunits of G proteins via these molecules, it leads to the activation of these targets, and this is what increases migration and proliferation of these tumor cells, and also provide more nutrients via angiogenesis, and it also degrades the extracellular matrix. And there is a number of biomarkers that can be established to diagnose cancer early and help develop cancer preventative treatments. Now, if we want to look more specifically with the CAMP and PKA signaling, some cancer types, it provides a tumor suppressive role, so that it prevents the production of tumors. And in other tumors, it produces a tumor promoting role. Now, how? Because the CREB, which is one of the targets of PKA, that helps promote invasion and metastasis. And a focal adhesion kinase, which is a type of enzyme, it also helps mediate cancer metastasis by CAMP. Another target of PKA is the CAMKK2, which stands for calmodulin-dependent protein kinase 2. And it phosphorylates and it inactivates this enzyme. And the CAMKK2 originally plays a role in energy homeostasis and insulin signaling. So let's look at more specific cancers that are caused by dysregulated GPCR signaling pathway. So examples of cancers are endocrine, thyroid, so anaplastic, also ovary, gastrointestinal, prostate, bladder, glioblastoma, atypical fibroadenoma, colorectal, chronic lymphoblastic leukemia, pleomorphic dermosarcoma, and hepatocellular carcinoma. So that's like liver cancer. So there's a variety of different cancers involved, and to cover each and every single one of these lectures won't be put justice in terms of looking at the causes, the effects, and so on, but we will provide a small insight in some of these. So hepatocellular carcinoma. So this is, in other words, a liver cancer, and one of the main causes in approximately 81% of cases is involved in hepatitis B and C. And when there is more damage in response to chronic hepatitis, this is what leads to cirrhosis. So there's more damaging, so there's more liver cell death. So this can lead to muscle cramps, weakness, nausea, vomiting, and the liver not functioning appropriately. And it can also affect other parts of the structures near it, such as the biliary duct. And amongst the main causes of cirrhosis is, for example, injury or alcohol, so increased consumption of alcohol. And that is essentially what leads to hepatocellular carcinoma. So in association with GPCR and HEC, inhibition of the enzyme CAMP-KK2 protects against HEC induced by a high-fat diet. Okay. Also, you have fibrolamellar hepatocellular carcinoma. So this is a form of a rare type of cancer that primarily occurs within children and young adults. And when there is a fusion, i.e. a binding between DNA GB1 and PRKACA gene, which normally occurs within 80 to 100% of cases, this is what leads to a removal of a gene fragment in chromosome 19, more specifically 400 kb, so kilobase gene, and also the production of a protein that helps retain the protein kinase A activity. So this is the link between liver cancer and the GPCR. Now, hepatitis B virus increases the risk of liver cancer, and more specifically, you have HBV X protein that can promote liver cancer, and this is via Creb MIR3188 and ZHX2 notch signaling pathways. And because Creb is part of the signaling pathway we discussed today, this is why it's been mentioned. And this is what increases the cancer cell growth and the invasion there. Now, with brain cancer, the main causes of this is, for example, with age, gender, and many other aspects. And especially for glioblastoma, it's mainly can be caused by radiation. And amongst the symptoms that you can have is buildup of fluid. There's also increase in pressure, intracranial pressure, many other symptoms. And in terms of the GPCR pathway, which we're focusing on, is that in glioblastoma, activation of pKa is what increases the CAMP levels, and it upregulates the PT1 and P27 genes, and this is what increases the growth of the cancer within the brain, and also differentiation and apoptosis, so the cell death. Now, within medulloblastoma, so this is a rare type of cancer. So this can affect adults, rare in adults, but more in children. And it mainly affects the location within the brain called the cerebellum. And in association with the GPCR, the enzyme adenyl cyclase that is found within the pituitary, that inhibits or prevents proliferation, and this is via the pKa GLI1 pathway. So you can see that the pKa CAMP signaling, it has the ability to either promote cancer or suppress cancer. Now, with lung cancer is like the second most common cancer within the UK, and there are many key factors involved, such as smoking, asbestos, if you work in environments such as factories and so on, and radiotherapy. There is also many genes that are involved in terms of lung cancer, where if they get mutated, it can lead on to lung cancer, such as EGFR, which we're looking at next week, the RAS, P53, also when you have mutated proteins involved in apoptosis, such as BCL2, and proteins involved in angiogenesis, such as VEGF. So when you have mutations in these type of proteins, that is what leads to lung cancer. So non-small cell lung cancer, NSCLC, this increases the CREB expression and phosphorylation within tumor tissues, and this is what lowers patient survival. And the CAMP can also lower apoptosis. Now, within small cell lung cancer, which is another type of lung cancer, you have an increased CREB expression, which increases proliferation, and also the CAMP-PA-CREB pathway could regulate the hypoxia response in lung cancer cells. The pKa increases cell migration and invasion. pKa induces protein phosphatase 2, phosphorylation and AP1, so this is what increases apoptosis. So over here, there's a variety of different types of lung cancer. So that depends on the position. So you've got the small cell carcinoma over here, and then you've also got the large cell carcinoma over there. So I don't want to go too much in depth because this requires another lecture itself. Now within the prostate cancer, inhibition of the ZAMPKK2 protects against prostate cancer, and progression of prostate cancer can occur through the pKa. How? Because the pKa subunits RA alpha of expression is linked with metastasis. So this is why it can be a suitable biomarker for cancer treatment by radiotherapy and chemotherapy. Also, overexpression of PKRI alpha is also associated with poor response to the radiotherapy. And testosterone, which is like a male hormone, it stimulates GPR56 directly, and this is what activates CAMP-PKA pathway. And this leads to another signaling pathway, which is called the androgen receptor. And this is needed for how prostate can progress in terms of cancer. So prostate is a gland that is found within men, and you can normally find it just below the bladder, and it secretes a fluid. So that is involved within the reproductive system there. And with ovarian cancer, so ovaries is where the eggs is being produced. It's also where the female hormone estrogen is being produced. So this is more or less involved in how the female are able to to break things down. Estrogen is the female hormone that is involved in menstruation. It's also involved in how the female develops during puberty or teenage stages, and also during pregnancy. Whereas the eggs is what being produced within an ovary, and this is what needs to be attached to the reproductive cell called the sperm that is found within the male reproductive system, and they combine together to form a fertilized egg, and that is what essentially grows within the womb to actually produce the baby. And pKa RI alpha is highly expressed within epithelial ovarian cancer, so that's within the lining, and it promotes invasion and metastasis by phosphorylating this protein called claudin 3, and this is what lowers the intensity or the strength of the tight junctions there, and it helps degrade the extracellular matrix. Now, when there is low CREB expression, this is what decreases the proliferation, but it has no effect on apoptosis. So here you can see the ovary there, then you've got a tube called the fallopian tube or the oviduct, and that is what leads to the womb. Now with colorectal cancer, PKA-RI alpha and ACAP10, they mainly increase tumor progression and metastasis, lowering survival. Also, there's a type 1 insulin like growth factor receptor IGF-IR signaling, which stimulates the phosphorylation of a protein called esrin. Now, when you have this protein esrin being phosphorylated, that is what leads to the CAMPA signaling and increases the tumor survival. Now with breast cancer, so breasts, these are the mammary glands that produce the milk via the structure called lobules. So this is where the area where milk is being secreted, and then they join together to form structures called ducts. And you can see with this image over here, you've got the normal ducts, and then there's growth and growth and growth until it forms metastatic. So what causes the growth of metastatic is the PKA, especially in a type of cancer called triple negative breast cancer. And this is via another signaling pathway called GSK beta-catenin pathway. PKA can also cause resistance to chemotherapy and, for example, trastuzumab, and this is present within HER2-positive breast cancers. And PKA-RI subunit also increases proliferation in normal cells and breast cancer metastasis. And the GPCR does increase the reprogramming of the cellular metabolism, so and how glycolysis takes place via the PKA-Creb pathway. So this is necessary to help facilitate growth of the cancer. Now with leukemia, what is leukemia? So leukemia is the overproduction of white blood cells, and it can also be in terms of the change in the shape or the mature white blood cells. So this is what suppresses the normal production of the white blood cells, and it can lead on to infection, for example neutropenia. It can lead on to bleeding, and it can be subdivided into acute or chronic, so that depends on how it's being progressed. Other symptoms can include enlargement of the spleen, the liver, can also affect many other sites. So in association with the GPCR signaling pathway, overexpression of Creb is found within the bone marrow of most leukemia cell lines. For example, B cell, which is a type of white blood cell. In the chronic state of this type, PDE4 inhibitors, so the phosphodiesterase that inhibits the CAMP production, induces apoptosis by increasing CAMP levels. So when you have inhibitors of this phosphodiesterase, that is what increases apoptosis because the CAMP levels are increasing, and it also blocks another type of receptor called toll-like receptor. And TLRs are a major role in innate immune responses. So when you have this protein called chemokines, so CXCR and CXCL12, produced within those white blood cells, they can bind onto the G alpha I subunit of GPCR on cellular cells, and as a result of that, that lowers the CAMP synthesis, because the G alpha I and the G alpha S are involved in the regulation of adenyl cyclase. So the G alpha I is what halts the adenyl cyclase activity. So as a result, you have low CAMP synthesis, and that is what increases tumor survival. So this poster I wanted to add in because it's very simplified and informative at the same time. So in acute lymphoblastic leukemia, this is what is found within lymphoid cells, so it can grow quickly, common in children, approximately 6,000 cases a year. Now, with the acute myelogenous leukemia, that is found in another type of cells called myeloid cells. So it can grow quickly, common in adults and children, and there's about 18,000 cases a year. Now, with chronic lymphoblastic leukemia, that is also found within lymphoid cells, and it can grow slowly instead of quickly, and it's common in adults over the age of 55, and there's approximately 15,000 cases a year. And chronic myelogenous leukemia, this is found within myeloid cells, that also grows slowly, and it's common in adults, and there's approximately 6,000 cases a year. So lymphoma. So lymphoma is another type of blood cancer, and you can have enlargement of the lymph nodes, you can have coughing, night sweats, itching, enlargement of the spleen, bone marrow involvement, because that's where production can take place. Also liver enlargements, and can affect the kidneys. Now, in association with GPCR, CAMP-PK can increase expression of BAX, which is an apoptotic protein that increases apoptosis, and it lowers the expression of anti-apoptotic proteins, which is BCL2 and survivin. So in other words, it can increase the rate of apoptosis there. So by the end of this lecture, what you should understand is GPCR are helical transmembrane receptor proteins found on the cell surface. It has 3 domains: extracellular, transmembrane, and intracellular. The activated receptor activates G protein, and how? When you've got an exchange between GDP for GTP. The G proteins are classified based on the alpha subunit. The adenyl cyclase stimulates the production of CAMP from ATP. CAMP then stimulates the protein kinase A, made up of 2 catalytic subunits and 2 regulatory subunits. CAMP is what binds onto the regulatory subunit of pKa. The CAMP response element binding protein is a transcription factor and one of the pKa targets, and it regulates activity of target genes to initiate cellular response. So that could involve a lot of, for example, growth, metabolism, angiogenesis, apoptosis, and dysregulated GPCR is one of the causes of this is point mutations. And the signaling pathway itself has tumor suppressive and tumor promoting roles. So increasing the pKa and CAMP does increase proliferation, differentiation, cell migration, invasion, and be able to evade apoptosis in many cancer types. And here are reference lists for further reading. Please do get in, if you've got time, do have a read of these. It's interesting. Next week, hopefully, we'll progress on to the EGFR. For any terminologies that you don't understand, please go and to the glossary to gain a further insight. I hope you enjoyed the lesson, and if you have any questions, please do not hesitate to message. Take care and see you next week. Bye.